A pleasant afternoon to all. It is my great pleasure to welcome you to the opening of the OMICSena lecture series. Some of you might have attended first OMICSena last March. That was an information field premiere of the OMICSena platform where DOST-FNI featured the frequently asked questions on Nutritional Genomics. For the second time, DOST-FNI brings you OMICSena with a bolder move to bring together omics experts from way beyond the Philippine borders. The Philippines continues to build strong innovative foundations, and learning and reflecting from the global scene is a very way to go. As such, we are thrilled to bring you the 9-day OMICSena lecture series. For 9 afternoons, we are expecting to learn the best of omics technologies and how these emerging platforms can enhance nutrition and food research that will eventually be translated for societal benefits of our country. Beyond basics, hopes are high that we can also build networks and collaborative paths, all towards building an omic-driven, innovative Philippines. William Wordsworth said that life is divided into 3 terms: that which was, which is, and which will be. So let us learn from the past to profit by the present, and from the present to live better in the future. I hope that through all the topics to be discussed from this day until October 7, we can learn how to create a better future in life and nutrition through the studies of omic sciences. To all the participants, again, my warmest welcome, and lend us your active participation because we have inspiring lecture series ahead. So thank you so much and stay tuned. Maraming salamat po at magandang araw muli.
Magandang agham po sa ating lahat. OMICSena was coined by DOST-FNRI to create a scene in a good way. It seems that it will continue to serve as a platform for the DOST-FNRI to keep us in the loop on its undertaking in genomics. With Dr. Brian Gonzalez on board, the second OMICSena for 2021 collaborates with the DOST's Balik Scientist program to drumbeat the advent of another set of omic science at the Institute, the Metabolomics. This expansion in the R&D platform will guide DOST-FNRI in setting up more actionable strategies in the molecular food and nutrition arena. I am optimistic that the series will provide new learnings to bring stronger and bolder omics-based research and development initiatives, not only in DOST-FNRI, but in the entire country, as I understand that this OMICSena is well represented by genomics enthusiasts mula Luzon, Visayas, at Mindanao. As we come together for the next 9 afternoons, I hope that we will be able to lay out plans and strategies towards the creation of new intervention systems, biomarker discovery, and innovative health and nutrition platforms through the use of omics technologies. May this event sharpen our vision to acquire the lens of seeing through what our country needs to reach zero hunger and optimum health and nutrition at the molecular level. Congratulations to the organizing committee of the OMICSena lecture series and to all the staff of DOST-FNRI led by Dr. Imelda Angeles Agdepa. Magandang agham sa ating lahat.
A pleasant day to all. The Nutritional Genomics program of the DOST-Food and Nutrition Research Institute is part of the DOST's Big 21 in 2021. This achievement highlights the growing field of nutritional genomics and the DOST-FNRI's New Gen Laboratory now an ISO 17025 certified molecular laboratory. Beyond this recognition, I am more thrilled to see what our R&D program and scientific and technological services in nutritional genomics can offer to ensure optimum nutrition for everyone. Earlier this year, the first OMICSena answered frequently asked questions on Nutrigenomics. The first installment of this web-based learning platform of DOST-FNRI's genomics programs made us realize that nutritional genomics can scale up nutrition and health sciences. Studying our genes through the omics platforms can bring about enhanced nutrition recommendations and perhaps nutrition and health policies in the long run. Its eventual integration in clinical practice can boost our efforts to overcome malnutrition and help us achieve our goals of good health and well-being. As DOST continues to expand the country's omics footprint, this second offering of OMICSena with Dr. Brian Gonzalez, our balik scientist for Metabolomics, is anticipated to bring new frontiers in omics-based innovations. This will fine-tune our efforts to use various molecular platforms to pacify and eventually end our long-standing battle against malnutrition and non-communicable diseases. Now, with the generous sharing of information and practices from our keynote speakers ahead of us, I invite you all to join me in getting a detailed glance at omics technologies and how they can play critical roles in nutrition and health. May this deep appreciation of omics technologies guide us in pushing for more innovations directed to our local needs. I am optimistic that this lecture series will also strengthen the competencies of our researchers in initiating omics-based research and development. One thing that the ongoing pandemic has taught us is to hold tight to what science and technology can do. Science and technology will always be an important key in solving various societal problems. Napaghahandaan man o hindi, katulad nitong pandemya, innovations and discoveries will help us thrive and become resilient. On behalf of the Department of Science and Technology, I would like to thank our speakers from different parts of the European Union and the United Kingdom for their time and expertise. I extend my appreciation especially to our DOST Balik scientist, Dr. Gerard Brian L. Gonzalez, for bringing this exciting series to light, together with the DOST Food and Nutrition Research Institute. Thank you all for being here. Let us have a learning-filled week ahead. Mabuhay!
In the food industry, ensuring the quality and safety of consumer products is always the priority. To meet this need, food manufacturers frequently rely on food testing laboratories to determine the nutritional content of food products and to examine its possible microbial and chemical contaminants to comply with regulatory requirements. Philippine regulatory bodies take the lead in ensuring that local laboratories have the capability to generate internationally acceptable analytical data. One of their bases in evaluating the competence of laboratory is the result of their continuous participation in Proficiency Testing scheme. Proficiency testing is an effective tool to determine the performance of a laboratory through an inter-laboratory comparison. DOST-FNRI, through its Proficiency Testing Laboratory, provides affordable, accessible, and reliable PT programs to support testing laboratories in their ISO 17025 accreditation. From 2006 to date, PT rounds were organized based on international standard, which includes PT on physical-chemical analysis on different food matrices. These FNRI PT rounds were participated in by 20 to 70 local and foreign laboratories. Seminars, trainings, or workshops are also organized after every PT round where participating laboratories are provided with knowledge that may possibly help them to improve their laboratory performance. Currently, the Proficiency Testing Laboratory of the DOST FNRI is the only ISO IEC 17043-2010 accredited proficiency testing provider in the Philippines, and is able to sustain and continuously expand its accreditation for 8 years. Aside from providing PT schemes, the laboratory also offers reference materials generated from PT rounds, which can be used for quality assurance purposes. Announcements on how to participate in the FNRI PT rounds, results of the current PT round, available reference material of the FNRI PTL, and their future plans are all made available in the IFNRI website.
In 2010, DOST-FNRI revolutionized its take on nutrition by incorporating genomics in its researches, investigating the relationship of anthropometric, biochemical, clinical, dietary, and health components to the genetic makeup of Filipinos. DOST-FNRI is looking into providing a more personalized approach of achieving optimum nutrition to every Filipino. Through nutrition, we attempt to better understand how a single nucleotide, a single letter, can make a big difference in our way of living. And so, to support this endeavor, DOST-FNRI capitalized on establishing its molecular laboratory known as the Nutritional Genomics, or the NewGen Lab. With its thrust of bringing science for the people, the NewGen Lab provides assessment of genes and genetic variants associated with micronutrient deficiencies and diet-related non-communicable diseases. The NewGen Lab is equipped with some of the state-of-the-art equipment in molecular biology, such as the automated nucleic acid extraction machine, bioinformatics, real-time and digital PCR systems, automated liquid handling system, micro-volume spectrophotometer system, and gel documentation system. With a complete workflow adhering to the highest standard of competency in laboratory testing, NewGen Lab is setting the mark in providing mole biotechniques with transparency, validity, and reliability. Whether you are an individual or a group of enthusiasts seeking answers in nutritional genomics, the NewGen Lab awaits you. Our proficient personnel works in close contact with our customers through customized analysis, provision of technical consultancy in nutritional genomics, and standardization or laboratory competency. To even serve you better, we are partnering with a leader in genomic testing for a higher throughput and faster turnaround time. Welcome to the new generation of nutrition. Welcome to the NewGen Lab.
Sorry. Good afternoon, Philippines, and good morning to the Netherlands and the United Kingdom. I am Diana Glades Tumalanta Ronquillo, and I am your moderator for today. Thank you for the warm messages from our Director 4 and Scientist 2, Dr. Imelda Angeles Agdepa, Undersecretary Rowena Guevara, and Honorable Secretary Fortunato de la Peña. Welcome to our Day 7 of the OMICSena Lecture Series. To our participants who are joining us every afternoon, starting Day 1, it is our pleasure to be with you again today. 2 more days before the series ends, we hope you have gained additional information and inspiration from our speakers. This OMICSena Lecture Series is a part of the countdown for the 75th DOST-Food and Nutrition Research Institute anniversary. Come and join us to celebrate the Diamond Year of the Institute on July 1, 2022. For 75 years, ang DOST-FNRI ay kasama niyo sa bawat yugto ng buhay at kabuhayan. To our participants, we are also live stream in DOST-FNRI YouTube channel. If you missed the previous episodes, you may still watch it at your most convenient time. Also, the attendance link will be provided in the chat box. As we continue, our speaker for today's lecture is Professor Jonathan Swan. He will be introduced by our Balik scientist, Dr. Gerard Brian Gonzalez. Over to you, Dr. Brian.
Thank you very much, Dayan. Magandang hapon po sa ating lahat at maraming salamat po sa inyong patuloy na pagsubaybay sa ating OMICSena Lecture Series. This afternoon, it is my pleasure to introduce Professor Jonathan Swan. Professor Swan is a professor of metabolism at the Faculty of Medicine at the University of Southampton in the UK. His research group specializes in the application of nuclear magnetic resonance and mass spectrometry-based metabolomics to study the mammalian supraorganism. He does this by disentangling the biomolecular contributions of the host genome, bacterium, virome, and the microbiome. He has specific interest in how these interactions contribute to the holobiont development and health, and how they are perturbed by nutritional deficiencies and infections. In addition, he has several ongoing projects studying the microbiota, gut, and brain axis. On a personal note, John and myself, we met a few years ago in Jamaica, where we had a meeting on a project on how to disentangle the molecular mechanisms driving increased risk for non-communicable diseases of children suffering from childhood malnutrition. He's also involved in several projects on child malnutrition and early life development. Mga kaibigan, it is my pleasure at kinalulugod kong ipakilala sa inyo, Professor Jonathan Swan. John, take it away.
Great, thank you, Brian. And thank you everyone for inviting me to talk today. I'm just gonna try and get this set up. So Brian, I'm gonna, hopefully you can tell me if you can see, if you can't see my screen, let's do it like that.
So far, so good.
Great, thank you. Okay, and then the laser pointer. Great. Yeah, so thank you for inviting me to speak today. And what I plan to do really is to introduce NMR spectroscopy, in particular how we use this for metabolomics, particularly untargeted metabolomics, and how we can use this to study how early life malnutrition can disrupt the biochemical system, and how that might then have an impact on health and development and potential diseases. And I saw some of the earlier talks. Brian gave a really good talk earlier on mass spec, and it's not really that people just do mass spec or just do NMR. Actually, more and more labs now do. The two techniques are quite complementary. And so, yeah, you often see labs applying both, but some labs only have one or the other. So just to start with then, I'm sure you probably had lots and lots of slides like this over the last couple of weeks, but traditionally, studies will have focused on measuring a small number of variables, a small number of metabolites, or perhaps a small number of proteins. And this is very much guided by the literature. And these traditional approaches then are heavily reliant upon a hypothesis, very hypothesis-led, based upon what we already know. And this can be quite restrictive. So what metabolomics tries to do, it tries to measure lots of, I think my computer is lagging a bit. Has been anything? Okay. Okay, yeah. So what metabolomics tries to do is measure lots and lots of information in a single measurement or a small number of measurements. And so by measuring lots of metabolites, we can rather be very focused, asking very specific questions. We can ask more open-ended questions. We can say, of all of these metabolites, which ones change with a disease, which ones change with a nutritional deficiency? And so this can be really useful for identifying something you wouldn't normally think of looking at, and be very useful for hypothesis generating. So rather than being led by what you already know, you can formulate new hypotheses, which you can then test with more traditional approaches. And we tend to use this to understand mechanisms of disease or mechanisms of, say, malnutrition, but also for biomarkers to identify people either with a disease or at risk of a disease. And another strength of metabolomics, like I say, you probably had lots of these talks already. Why my slides aren't shifting. Okay. Is as well as seeing with metabolomics, this is an NMR spectrum here, as well as seeing metabolites that come from the genome, so endogenous metabolism, metabolomics also has the benefit of seeing signals from the environment. So we can detect in this case, again, it's an example of an NMR spectrum, we can detect signals that come from dietary intake. So whether it's the dietary intake themselves or the digestion products of those different inputs, those different nutritional inputs. And we can also see metabolites that derive from the microbes that live in our body. And so using metabolomics, we can gain this overview of the whole metabolic system, not only the human side, but also the microbial contribution and how it responds to the nutrients. Sorry, for some reason my slides aren't shifting very well. Right, have other people been having this problem, is it? An easy fix. Okay, there we go. And we can also see metabolites that arise from interactions between these. So I thought what I'd do is for sort of 10 minutes, I'd just introduce NMR spectroscopy and the theory behind it. Now this is very much a sort of surface level overview. So it's no means kind of a complete deep explanation, but really we apply NMR in what we call an untargeted manner. We just measure everything we can see, and in particular for metabolomics, we're interested in proton or hydrogen NMR, and so we're just trying to measure all those metabolites that might have a hydrogen on them. I'm going to regret putting so many animations in. There we go. Okay, so with NMR spectroscopy, we're concerned with measuring hydrogens. And so if you think of a hydrogen, you could think that at its nucleus, we have one proton and no neutrons. And so it's positively charged nucleus. And when it comes to metabolites, if you think the majority of metabolites we're interested in, so this is just an example of all the amino acids, you can see that most metabolites contain hydrogens, and they contain hydrogens in slightly different chemical environments. But one thing, just take my word for it because we don't really have time to go into it, but these hydrogens have a property called spin. So they're all spinning. So all these hydrogens and all these metabolites are spinning. And because they are spinning and they have a positively charged nucleus, this means that they have a property, or they have a magnetic moment. Yeah, because they're spinning and they're positively charged, they have a magnetic moment. And when it comes to hydrogens, hydrogens have two possible spin states. They have a low energy spin state and a high energy spin state. And what this means, so all those hydrogens and all those metabolites I showed are all spinning, and because they're all positively charged nucleus, they all have this magnetic moment, and they can be present in either a high energy spin state or a low energy spin state. And what this means is when we put them in an external magnetic field, like an NMR spectrometer, they align either with, if they're in the low energy state, they align with the magnetic field, or if they're in the high energy state, they oppose it. So if you imagine you've got your sample, let's say a urine sample with all these different metabolites in that have all got hydrogens on, all of those hydrogens are randomly spinning in different directions. You put them inside the NMR magnet, and then they align either with or against. Now you have both these high and low energy protons, but you always have a slight excess in the low energy state. And so some of those low energy protons will cancel out the high energy protons, but there's a slight surplus, a slight excess in this low energy state. And this is the signal that you measure. And so what we do, we have this low energy state protons, and when we add in energy at a particular frequency, we irradiate those nuclei with this particular frequency of energy, it will cause them to flip from the low energy state, will absorb that, and flip from the low to the high energy. And it's this transition between the two energy states that we're measuring. And when they flip, they are what we say they're in resonance with that applied radiation. And so with NMR, so it's nuclear because we're measuring nuclei, magnetic because we're exploiting this magnetic property, and resonance because we're measuring when they resonate, that frequency of energy. Now, the important thing here is that not all protons have identical resonance frequencies. So hydrogens, protons on different metabolites, or even hydrogens on the same metabolite, they're in a different chemical environment, will resonate at a slightly different frequency. And so this is kind of the fundamental of how we use it for NMR-based metabolomics. And the reason for this is that you remember you have this proton, this positively charged nucleus that you're measuring. And this experiences this magnetic field, but this is influenced, the magnetic field it experiences is influenced by this electron, this orbiting electron. And this shields this nucleus, something we call nuclear shielding. Now, if you put that hydrogen on a carbon atom, it's going to pull that electron further away from the nucleus, de-shielding that nucleus. And the chemical environment that hydrogen's in, like the electronegativity, is going to change the shielding of that hydrogen, that proton, and that's going to change the frequency at which it resonates. And this is essentially how NMR.
We may have lost you, John. John, are you still there? Okay, yeah, you're back.
Okay. Yeah, I'm good. Okay. What might do? Just give me 1 minute. Try and is my voice stable? Is that okay, Brian? Just give me a thumbs up. Or...
Yep.
Yeah. Okay. So I don't know when I dropped out, but essentially, hydrogens in different chemical environments will be shielded or de-shielded to different extents, causing them to resonate at slightly different frequencies. And that difference in frequency so this is an example of an NMR spectrum. And the position along this x-axis is what we call the chemical shift. And this reflects, this shows that difference at which the frequency at which they resonate. So signals that come down here are protons that are shielded. Protons that are down here that produce peaks down here are very de-shielded. And so we can use this position on this x-axis to understand information about the chemical environment, and we can use this to understand which metabolite that peak that we're seeing belongs to. And so this is the example of a human urine sample. This takes about 5 to 10 minutes to acquire. And so that one of the strengths of NMR spectroscopy is that it's very quick, it's fairly minimal sample prep. It doesn't destroy the sample, so you can reuse the sample for other things. And it's very robust. So we just, for example, we've just done a big run looking at 3,500 urine samples, and it's with QC samples, you can see it's very stable across that long run. So that run took, I think, a month of NMR analysis. And so it's very stable and reproducible. And what we see here then, each peak corresponds to a hydrogen in a different chemical environment, and the height is proportional to how much of that hydrogen is there, or how much of that metabolite is there. So in 5 to 10 minutes, we've acquired the metabolic profile of this sample, this urine sample. And we can use NMR for urine, for blood, for fecal samples, for tissue samples as well. And the sort of a major strength of NMR is that, like I say, it's reproducible, but this, if we run this urine sample here in Southampton, then this metabolite, for example, lactate, will give the signal here. But if we run the same sample in the Philippines, it will give the signal for lactate at exactly the same position. It will look the same. And this is really good for making databases, which can then help with the metabolite identification step. And so that could be one of the bottlenecks for, particularly for mass spec, and sort of this is a slightly easier part for NMR. Also the fact that we can use this pattern, these NMR signals, to understand the chemical structure of the metabolites as well. So the idea is here, you've untargeted, you've measured all the hydrogen containing metabolites in a sample. And then you want to get that meaningful information out. And so I thought I'd just kind of introduce this slide, because I think Brian might be giving you a tutorial on data analysis maybe next week. And so here, so we have this spectral image, this NMR spectrum of, let's say, a urine sample. Well, this is made up of lots and lots of data points. And actually it's about 20 to 30,000 data points make up this NMR spectrum. And so how we convert this spectral image into some data is we digitize it. So here, for example, every single data point becomes a column. So these are our variables, and the name of the variable relates to the chemical shift. And the value we give here is the intensity value, or the height of that particular data point. And so each person's NMR spectrum becomes a row of data. So here in this case, we measure 25 urine samples, 25 people, 25 observations. This becomes, we have 25 rows. And so just for some notation, we tend to refer to this as our X matrix, where we have n rows for n observations and k columns for k variables. And so in this example, we've got 25 people, 25 rows, and we've got 20,000 data points. And so we use that complete spectral data for our input. And so this is very much the untargeted manner. And so we put all of that into our data analysis, our multivariate statistics. And so at this stage, we don't know what this peak is. It's only when we run the analysis and we find that this might be related to growth or to intake, that we then go and try to identify what it is. So you've got that mass of information, that mass of data. And so, like I say, there is data within that might relate to nutritional status, psychological stress, pollution, environment. We want to pull out meaningful information. And so to do that, we apply a whole range of different multivariate statistical approaches. And this is just the most basic approach, and you've probably heard about this already. This is principal components analysis. And so what this does is it takes that those 20,000 variables, that full spectrum, and it summarizes each person's metabolic profile as a single point. So each triangle here is one person's urine metabolic profile. And PCA is very basic. It just looks at variation in the data. And so the way to interpret this is that points that are close together, like these two, they have a very similar metabolic profile. And points that are far apart are more different. And so with the right study design or the right metadata, we can identify vectors within this mass of information. So for example, if we know that these are poorly growing children, these are good growing children, we can identify this vector, then we can look at which metabolites change as we move along this vector. We can then take that information out and we can formulate hypotheses on that. And we can also look at relationships between metabolites as well. And so what we tend to do is when we find signals or peaks that are significantly associated with different bits of information, we can integrate those metabolite peaks. And if we integrate the metabolite peaks in the NMR, they are effectively relative abundance. And so we can then come down to these more defined variables. And then we can look at relationships with positive or negative between these. And this was just an example in some children from Malawi, where we found that tryptophan was strongly related to lots of these pathways we were interested in. There's things like gut health, tryptophan metabolism, and muscle metabolism. And so I identified that tryptophan might be a good intervention to target. So with that, I'll just kind of then, hopefully that's an overview of NMR and how we do NMR metabolomics. And then we can use this and we just give sort of 4 examples to sort of demonstrate this. Before, I've just got recording stopped. Before I go any further, is there any questions? Is everything clear on that? And can everybody still hear me?
Yes, John, we can still hear you.
Was there any specific questions?
I don't see any questions on the chat box.
Great.
That being said, if you do have questions, please type them in.
Great. Yeah, I thought I could leave my chat box open, but for some reason it shut when I loaded the slides. So yeah, so I can't actually see any questions, but yeah, just feel free to, and then maybe Brian can ask them if needed. Okay, so to show how we use NMR then to study undernutrition. Now, I think most of the people on the call are probably quite familiar with this, but I'll just introduce it anyway in case there are people that aren't. This is this idea of the vicious cycle between malnutrition and enteric infections and how this can impact on development. And there's this idea then that infants that suffer from malnutrition, particularly undernutrition in early life, this can impair their immune system and their defenses. They're often in environments where they're exposed to higher burdens of different pathogens or parasites. And this can lead to a greater number of enteric infections and also more persistent enteric infections. And in turn, this can lead to intestinal damage, inflammation, and which can lead in turn to malabsorption of nutrients and can also lead to diarrhea, which can lead to nutrient loss. And so combined, these things make the malnutrition even worse, which feeds this cycle even more. And the idea is if this is happening over the first few years of life, this can lead to things like growth shortfalls and stunting, cognitive impairment, and impaired vaccine responses as well. And then there's this double burden later life obesity as well. And so what we're trying to do, and Gordana spoke last week about the metabolic impact of enteric infections, we're trying to understand more and more what's happening metabolically throughout this cycle. And so this was actually what I'm going to do. I'm just going to, might be easier for you to see on this slide first, just to give you an idea of the NMR spectrum. So what we've done is we've looked in urine samples from children. So in here, this is children from Peru, this part of the MALI-D study. And here we were looking at which metabolites were related to growth. And in this example, this is change in length for Z scores over a 6-month period. And here, this is a PLS analysis. So what this looks at is metabolites or peaks that are significantly related to these measures of growth. And what we see is anything pointing up is excreted in higher amounts by children that subsequently go on to grow better 6 months later. Anything pointing down, they excrete in lower amounts, or to look the other way, anything pointing down is excreted in greater amounts by children that didn't grow very well. And it's colored by strength of that correlation, that significance. And so what we see quite clearly is that these two metabolites, betaine and this metabolite dimethylglycine, are strongly or excreted in higher amounts by the children that grew well. And we see the same thing in children from Bangladesh and Pakistan. I'm just going to go back to this slide. And so again, we also saw the same thing in Brazilian children. So in this study, we looked at around 300 children from Brazil, all under 2 years of life. And here, what we've done is just to simplify it and to ease interpretation, anything with a color is significantly associated with these different measures of nutritional or growth. So high for Z score, we're particularly interested for looking at stunted growth. And so if it's green, it's excreted in higher amounts by children that grew well. If it's red, it's excreted in higher amounts by stunted children that grew poorly. And so again, as I just showed for Peru, children that grew well excreted higher amounts of betaine and dimethylglycine. Now, you can get betaine directly from the diet, but it also comes from the breakdown of choline, where you get the majority from your diet. And so you get your dietary choline. This is broken down to betaine. These are really key methyl donors. And betaine gives up a methyl group to become dimethylglycine. And that methyl group helps drive this homocysteine methionine pathway. And this is really key for making this metabolite, S-adenosyl methionine, which I'll talk about in a minute. So like I say, we see the same thing in Peru, Bangladesh, and Pakistan. And what is driving this? This is, like I say, we see this in lots and lots and lots of studies. And choline is really important, methyl donor, it's also important for growth. And what we think is driving this lower choline in the stunted children, we think, one, they're probably getting lower amounts in their diet anyway. But also we've got data showing that the malnourished microbiome also kind of competes with the host. It has an increased preference for this choline in the gut. And so it converts it to trimethionamine and dimethionamine. And what this does is it lowers the bioavailability of choline for the host. And we've got some data, I think Gordana showed last week, that Cryptosporidium in particular can also consume, it can compete with the host for this dietary choline. And so this lowers the amounts available for the host. And in a few slides time, I'll show you, we also got some data showing that gut damage also prevents this absorption. And so the consequences for this is there's less choline entering the body, and less betaine as well, and less betaine then available to drive this pathway. And this pathway is key for making S-adenosyl methionine. And so S-adenosyl methionine is the body's primary methylating agent. And this is really important for DNA methylation. So SAM brings these methyl groups to this reaction, and this methyl group helps to tag genes. And when it tags genes, it helps to turn off their expression. So you're obviously born with a static genome, but this epigenetic process of DNA methylation is really important early in life for fine-tuning your genome to match that of the environment. And this is really important, this is particularly important in early life, and it's really important for development. There's some data showing this is really important for brain development and brain function. and it's heavily dependent on the availability of SAN, which is heavily dependent on the availability of choline and betaine, which we see lower in these children with poor growth. And so this raises the potential, can we supplement choline to try and improve this? And choline, eggs in particular, are particularly rich in choline, and people have tried giving increased amounts of eggs to children and shown some, in some studies it's worked quite well, in other studies it hasn't worked so well. So the next area I want to talk about is the microbiome. And so again, I think most people are probably familiar with the microbiome, but just as way as introduction, the human body is now, the current estimate is that we're colonized by around 37 trillion microbial cells, and that's both in and on our bodies. And the best studied of these are the bacteria, and in particular, the bacteria are predominantly located in our gut, in particular the large intestine or colon. And what's important to remember then is each individual bacterial cell has with it an accompanying genome. Different bacterial cells then bring with them different genomes which contain different genes. And so if we think about all of these genomes collectively together, we often refer to this as the metagenome. You can start to appreciate that all of these different bacterial cells in your gut bring with them different genes, that collectively the genetic content of the metagenome is much bigger than the human genome. It's estimated that there are 3 million different genes in the metagenome. And when you compare this to the human genome of around 24,000 genes, you can see there's anywhere from 100 to 150 times more genes in the metagenome than the human genome. And many of these will encode proteins or enzymes which will perform metabolic functions. And you can start to appreciate then that the microbiome, the metagenome, really does expand our metabolic capabilities, our ability to process nutrients, different environmental inputs, as well as host secretions. And so this is why we often refer to the human body as this superorganism, which has this metabolic contribution from the genome, but also the metagenome. And so it's important to think about this when we're studying things like undernutrition, but nutrition even in sort of general. And the reason why I'm talking about this now is one of the main signals we see in stunting is we see that the stunted children excrete higher amounts of these metabolites, which come from the microbial breakdown, particularly of amino acids. So stunted children excrete higher amounts of these microbial metabolites. And so just to highlight here, we've got paracresol sulfate and phenylacetylglutamine. So just to show you how these are formed, so in the colon, we have tyrosine, which is an amino acid from the diet. This is metabolized, particularly linked with members of Clostridia, to paracresol. This is then absorbed from the gut, and this is fairly toxic for the host. So it's sulfated in the liver and excreted as paracresol sulfate. We also see phenylalanine is an amino acid that gets broken down to phenyl acetate. This is absorbed and is conjugated with glutamine to make phenylacetylglutamine. And again, we see both of these increase in the stunted children, suggesting that their microbiomes are converting more of these amino acids into these products. And so generally speaking, a healthy type microbiome metabolizes lots of carbohydrates into short chain fatty acids, which tend to have more beneficial effects on the host. Whereas the more proteolytic microbiome tends to produce these kinds of compounds, which have been associated with more toxic, more harmful effects for the host. And we see that the malnourished microbiome tends to be more proteolytic in its functions. And again, yeah, you see this then higher in the stunted children. And we and others have reported there's lots of human studies now showing that undernourished children, stunted children, wasted children, children with SAM all have disrupted microbiota, which is not surprising given that the microbiome relies on the host for its sort of nutritional substrate. And we've seen here, this is a mouse model here looking at on a nourished diet, you see this nice maturation of the microbiome. Whereas if you put them on a protein deficient diet, that maturation is delayed and then they eventually end up in a completely different space to the nourished. And metabolically, we see differences as well. And this was a study a couple of years ago now showing, so normally your microbiome is nicely compartmentalized. So you have an oral microbiome, a small intestinal microbiome, a colonic microbiome. And this is controlled by things like your immune system, but also physiological factors like pH. But what they observed is in stunted children, that compartmentalization is lost. They found members of the oral microbiome in the gut, and in particular, they saw the small intestinal bacterial overgrowth. So bacteria that normally restricted mainly to the colon had overgrown into the small intestine. And we think one of the drivers for this, so this is work we've done with Paul Kelly at QMUL. So this is in Zambian adults. And here what we looked at, so Paul collected aspirate samples from the duodenum. And so what we're looking at here, these are the NMR metabolic profile, the metabolites present in the small intestinal lumen. And in these same individuals, blood was collected and we measured this, this marker, intestinal fatty acid binding protein, or IFABP. And this is a marker of gut damage. The idea here is that your enterocytes produce IFABP, and when you get gut damage, this is leaked into the blood. So if this goes up in the blood, you've got damage to your gut, to your small intestine. And so we looked at which of these metabolites in the lumen were correlated, were associated with this measure of gut damage. And so anything pointing up is higher in the small intestinal lumen when there's gut damage, anything pointing down is lower. And what you see straight away is there's lots of amino acids are more abundant in the lumen when there's gut damage, as well as choline. And we think there's probably two things at play here. One is that with gut damage, you've got malabsorption. And so these amino acids and choline, which are normally absorbed in the small intestine, they can't be absorbed. And so they stay in the lumen. And this could also be leakage of these amino acids and choline from the enterocytes into the lumen as well. And as a consequence of this, then, these become available for the microbiome, the substrate for the microbiome. These become greater substrate for the microbiome. And so they stimulate the growth of the bacteria and the small intestinal bacterial overgrowth, which is probably what's driving that more proteolytic microbiome, as well as that increased preference for choline. It's selecting those bugs. And we've also got more recent data. This is in children, again, showing the same thing. This time using a random forest type approach. If it's green, it's positively associated with gut damage. If it's red, it's negative. So you see glucose goes down in the gut when there's gut damage. Whereas these other metabolites, they stay up, so they can't be absorbed. And this is consistent with what we see. So the stunted children, where you'd expect there to be gut damage, have higher amounts of these amino acid metabolites from the microbiome. And does this have a functional consequence? So the idea here is that if the stunted children have more paracresol sulfate in their urine, it suggests that their gut wall is exposed to higher amounts of paracresol, this microbial metabolite of tyrosine. So we did a transepithelial resistance test here, where you take CACO2 cells or intestinal cells, and when you grow them, if there's nice tight junctions between the cells, they're healthy, they have a lot of regulation. If you pass a current across them, you should get a high resistance. If those tight junctions between those cells is reduced, then the resistance goes down. So we took these CACO2 cells and incubated them in different concentrations of this paracresol. And what we found is that the higher dose of paracresol, we didn't see toxicity at this level, in the higher dose of paracresol, that resistance went down, showing that the microbial metabolite could actually impair these tight junctions between these intestinal cells. And we think this may contribute to a leaky gut phenotype, which is also seen in these infants. So it's funny because you have this sort of difference throughout the gut where you have malabsorption in some places and leaky guts in others. And this leaky gut as a consequence, because the host loses its ability to control what moves from the intestinal lumen into the systemic circulation. So the idea here is that if you have a microbiota that's producing lots of paracresol, it disrupts this gut barrier, allowing the translocation of bacteria, of pathogens, and in particular gram-negative bacteria. They have this cell wall component like a polysaccharide, or LPS, and LPS is a potent inflammagen. So this inflammatory signal can move into systemic circulation where it can trigger inflammation. And this has a consequence for activating this tryptophan kynurenine pathway. And so that, so that adds this, that leads us into this next pathway, the nicotinamide tryptophan pathway. And so this is slightly complicated, and Gordana kind of touched on this, so I'll try and go slowly through it. But essentially, inflammation is good in some ways because it helps the body to clear pathogens, but chronic and sustained inflammation can lead to damage that's unwanted. And so the body has this kind of compensatory strategy, whereas when you have inflammation, it activates this enzyme IDO, indolamine dioxygenase. So when it encounters inflammatory signals like interferon gamma or LPS, this enzyme IDO is induced. And what this does is this enzyme is the rate limiting enzyme for breaking down tryptophan into kynurenine. So when inflammation is detected, IDO is switched on, tryptophan is converted to kynurenine, which is broken down to 3-hydroxyanthranilic acid. And when this is increased, what this does, 3-HAA and kynurenine help to drive naive T cells away from the pro-inflammatory Th17 profile and move them towards the regulatory or Treg or more tolerogenic profile. So it dampens that inflammatory response. And so this is good because it stops that damage associated with chronic inflammation, but it's bad because it diminishes the host's ability to generate this Th17 response, leading to a loss of that mucosal immune barrier and increased susceptibility to infections. We think this may also contribute to that more persistence of pathogens as well. And so this is something we actually see. So you can measure tryptophan and kynurenine in the blood. And if you look at the kynurenine to tryptophan ratio, that gives you a measure of how induced IDO is. So the KT ratio is a measure of how induced IDO is. And we've actually seen there's data showing in MALI-D that stunted children have a higher plasma KT ratio. And we've recently shown in children from Zimbabwe in the SHINE study that stunted kids also have a higher plasma KT ratio. And so one of the consequences for this as well is the impact on nicotinamide. And if we go back to, this is the Brazilian study. So here, this last column, here we looked at children that were stunted. We measured their urine metabolic profiles, and we looked at, was there any signal in their metabolic profile that predicted how they were going to grow 6 months into the future? And so looking for potential markers of catch-up growth. And so again, anything in green is higher, excreted in higher amounts by children that catch-up growth. Anything in red is lower in the children with catch-up growth. And one of the signals we saw was this metabolite, NMND, or N-methylnicotinamide. And so NMND, N-methylnicotinamide, comes from the breakdown of NAD. So NAD is really important for energy generation, which is key for growth. And you get the majority of your NAD comes from tryptophan metabolism. And it's thought that if this is chronically activated, this IDO, it's going to dysregulate tryptophan metabolic pathways. Also, you've got lower amounts of tryptophan in the diet, this essential amino acid. And so this is going to have implications for NAD plus generation. And like I say, NAD plus is really important for energy generation, which is important for growth. But NAD is also used by a whole variety of enzymes such as sirtuins. And when they use NAD plus, they convert it to nicotinamide. And this nicotinamide can then be recycled back to NAD plus via the salvage pathway. Or you can make NAD plus from dietary intake such as nicotinic acid or niacin or nicotinamide riboside. But if you've got a surplus, if you've got an abundance of NAD plus, when this NAD plus gets broken down to nicotinamide, what the body does is it can methylate that nicotinamide and excrete it in the urine as NMND. And so we think this is probably a marker for an abundance of NAD plus. So what that shows is children with catch-up growth had plenty of NAD plus, whereas the children that didn't grow well didn't have much NAD plus. And so there's a variety of studies underway trying to supplement these pathways at different points to see if you can boost NAD plus and whether that can boost growth and health. And so we part of a study called the ILLICIT study, which was done in Tanzania, where one of the treatment arms was nicotinamide. And this was given to pregnant mothers and then also infants in early life. Unfortunately, spoiler alert, it had no impact though on growth, looking at LAZ, WAZ. So it's probably too simplistic just to give this single nicotinamide. Perhaps we also need to give it in combination with things like choline as well as other amino acids. But we're currently working through all this data now to try and understand what nicotinamide supplementation did do for metabolism, and is there anything we can learn from that? And one of the reasons why this may not have worked so well, or why we need to be a bit more elegant about the way we do it, is this idea of biochemical aging. Now, this was a study using the NMR approach on the Mali D cohort. So here we looked at children from Peru, Bangladesh, and Tanzania. And we had healthy growing children and that grew healthily across the full study, and those that were growth constrained, so those that had poor growth throughout the study. And we had urine samples and blood samples from these children at 3 months, 6 months, 9 months, 15 and 24 months of life. And so we could look at which metabolites changed with age. So was there this age-dependent signature of the metabolome? And so then we could use that to understand more about biochemical aging. And so to start with, we just focused on the healthy children and we pulled them from the 3 sites. So we could get this universal healthy signature of biochemical aging. And what you see here, anything with color is significantly associated with age. And if it's red, it's positively associated with age. It goes up as children get older. If it's blue, it goes down as they get older. And if we pull these children, we were then able to find about 22 metabolites, I think it was, that changed with age significantly. And you've seen some of these already. It's really nice because there's a whole range of pathways covered by these. And so what we wanted to do was, could we build a mathematical model where we could just feed in the metabolites, the metabolite abundances of these 22 metabolites to predict the age effect. And we referred to this as phenome age. Sorry, my slides are going a bit crazy. We refer to this as the phenome age. And actually, because we wanted to try and use this in the field, we refined it down even further to the 8 most influential metabolites. And you see here, this is their influence on this phenome for age score. And so we came up with these 8 metabolites. And so now the idea is we can take a child that's under 24 months old, we can measure these 8 metabolites, put the values into our mathematical model, and it will tell us the biochemical age of that child. And here we can then understand, is the child's biochemical age the same as their chronological age? Because if their biochemical age is younger than their chronological age, then it shows that they're biochemically immature. If it's higher, then it shows they're more biochemically mature. And this is really important because most interventions are targeted at the child's chronological age. And not all children, as we'll see, have the same, they're at the same position. Their chronological age does not necessarily reflect their biochemical age. And so, as is standard in this field, we standardize these into Z-scores. So we'd like to call it the metabolome for age Z-score, but MAS was already taken because there's a microbiome for age Z-score. So we called this the PAS, or the phenome for age Z-score. And so this allows us to then standardize and compare across sites, across children. And what we see is when we look at the PAS scores, then if we look in Peru, you can see at 3 months. So here we've got the darker bars are the healthy grown children. The light gray are the growth constrained children, those with poorer growth. And we see that even as young as 3 months old, there's almost 2 standard deviations lower in the growth constrained children compared to the healthy grown kids. So even at 3 months, they're already biochemically, and this is statistically significant, they're already biochemically immature. So if we're targeting interventions, we're probably already too late at 3 months. And we see this, and then they stay significantly different across the study. And where it gets really interesting is when you look at the individual metabolites. So 3 of these metabolites are metabolites of the microbiome, showing the importance of the microbiome in this superorganism development. And then we have we've got 2 metabolites that come from the TCA cycle, and this shows the importance of energy metabolism in early life. And actually nicotinamide was, NMND was significantly associated with age. It was strongly associated in Peru and Bangladesh, but less so in Tanzania. I think this is more to do with the fact that we had much lower numbers of children in Tanzania. But yeah, it's just an interesting observation there. If we look at length gain then of these infants, we can see that you have this huge amount of growth very early in life, and that slowly tapers off. And we know that around 35% of your energy, your total energy requirements, are dedicated towards growth in that first 3 months. And then this halves by 6 months, and it's as low as 3% at 12 months. And that's reflected quite nicely in these TCA cycles, particularly in citrate. So you can see that this first sort of 3 to 6 months, really important for having enough energy to sustain growth during this period. And again, if you're trying to target interventions to promote growth from an energy point of view, if you're sort of after 9 months, you're probably already too late. And when we look at the betaine and dimethylglycine that we introduced earlier, you can see that the betaine tracks almost perfectly that length gain curve. And again, it kind of reinforces the importance of those methyl groups in early life. You have to have this high abundance of these methyl groups during this sort of first 9 months. And then after that, it's too late. And again, choline, we also know is really important for muscle performance, muscle development as well. And we know it's also really important for brain development. And so I think this is really important that future, I mean, it's what's practical. Not everyone has an NMR spectrometer in the field, but it kind of emphasizes this importance of tailoring interventions to the biochemical needs of the child rather than their sort of just blindly their chronological age. And the fact that these things differ between good growing and bad growing children. So then, just to sum up then, I talked about NMR spectroscopy, nuclear because we're measuring nuclei, magnetics, we exploit this magnetic property, resonance because what we're measuring is how they resonate at different frequencies. We take a urine sample, or it could be blood or tissue sample. We have hydrogen containing metabolites that we measure their untargeted metabolic profile. We convert that into a data table, which can then be used in a whole variety of multivariate statistical approaches. We have this, so we have these inherent inputs like the genome, epigenome, immune system that drive the metabolic phenotype, but this is also shaped by the environment as well, like nutrition, the microbiome. Could probably put pathogens in there as well. And these are all interrelated as well. And so the nice thing here is we can actually tweak the microbiome or the nutrition, whereas we can't necessarily interact with these things. And this can shape the metabolic phenotype, which can drive health and disease. And we know that the metabolic phenotype, and actually all of these components, change throughout the life course, and they influence healthy development, later life outcomes. And we can tweak these inputs to enhance the metabolic profile to try and improve things like healthy development. So with that, I will acknowledge following people. I'm happy to take any questions. Yeah, and if not, if you don't want to speak now, feel free to email me afterwards. That's fine.
Amazing. Thank you very much, John. Yes, Diane.
For now, we don't have any questions. To our person, you may type in, yes.
Okay. Well, I have. Hi, John. While waiting for the others to put that in, I have to say that was the clearest explanation of NMR I've had. So that's very good. Just before I raise my questions, I just want to say that I believe in the Philippines, we have one NMR facility that I know, and that's in the DLSU. So De La Salle University has a 600 hertz NMR, I believe. So if you do have questions and biological samples that you are and you would like to explore NMR for your metabolomics work, you can contact Karen Lacerna. I don't know if Karen is here. Karen, are you around? I don't think so. Okay, yeah, but anyway, you can contact Karen Lacerna, and I'm sure myself, Gordana, and maybe John will be willing to discuss any interesting research you have in the Philippines. I guess my first question, John, is so for to clarify with everybody, of course, the difference with NMR and mass spec is that in mass spec, each feature normally, if you're lucky, you get one metabolite. If it's not noise, or if it's not, but it could also be an isobar. So one peak or one feature is one metabolite. But with NMR, of course, you have separate, you have different chemical shifts that represent one metabolite, right? So it works in the same principle as what an FTIR or something where each molecule has different peaks, in fact. So one molecule is represented by several peaks. So I'm just curious how, like in your studies, for example, you have like a heat map where the x-axis is betaine. How is that? How did you get that value? Like, for example, in this one, how did you derive at that value?
Yeah, so let's see if I can. That's bad example, actually. Good question. So, for example, choline, let's just say this is a singlet. Actually, no, let me pick one that has two. Let's just say lactate. Let's say this signal and this signal are the same metabolite. Then, there's different ways to do it. Now, they are proportional. So if, let's just say there's 5 times more hydrogens in this chemical environment on alanine, and let's say 1 times more hydrogen on this hydrogen on alanine. So if there's twice as much alanine, that's going to go up twice as high. That signal is going to go up twice as high. So it doesn't really matter which peak you take, because they should both move up and down to the same extent. What we tend to do, if we're identifying a single metabolite integral, is we select the peak that is not overlapped by anything else. So that's kind of the main driver, because you don't want, let's say there's a metabolite here and there's another metabolite underneath it, then that would be a bad peak to pick, because you're not getting purely that metabolite, you're getting that metabolite plus something else.
It would be rather difficult anyway to get. Well, is it very easy to get a unique shift per metabolite? Is there Is there
Yeah,
one
so,
unique
yeah,
peak for okay?
this is kind of where the field strengths come in, because you get more dispersion with the higher field strengths, and so as that happens, you can the signals separate more, and you can actually be very specific. Now, when it comes to, so there's two ways. One is that you express relative abundance, and so let's just say, I didn't really go into it, but the peaks split, so you can get singlets, you can get doublets, you can get triplets, quartets. Now let's just say you've got a triplet, because if you're doing relative abundance, you can just select one of those peaks on the triplet, because again, they're all proportional to each other. And so you can pick one that's not overlapped. What you can also do, because we use an internal standard TSP, some people use DSS, and you know how much you put in, and you know that the peak that comes from that TSP relates to 9. So let's say you put 1 millimolar of TSP in, and there's 9 hydrogens give rise to that signal. And let's just say, I don't know, this alanine peak, comes from 3 hydrogens. Well, then you can put all those into an equation to work out the sort of approximate concentration of alanine as well. So you can also, it's still really relative abundance. It's not it's not an accurate quantification, but you can get a sort of ballpark figure of quantification as well.
Well, on that point, we have a question on how do you do quantitative analysis in an NMR, because it's always sold as a very robust, very quantitative analysis, which I agree. But how does that happen?
So how the NMR is set up, say for example here, is that it's not, I wouldn't say it's quantitative in this approach, because it's optimized to capture lots and lots of information across the full spectrum. If you wanted to, let's just say, quantify alanine, then you would have to optimize your NMR parameters on this particular peak, this particular hydrogen. Now, that being said, it's not far away from accurate, but given the field we're in, analytical chemistry, you don't want to say that is a quantified metabolite. It's kind of a close approximation. You know that it's in the millimoles rather than the whatever. It's so that, really, in this style of approach, an untargeted approach, you can't get accurate quantitation. But there's nothing to say you can't go back and pick a particular sample and then quantify that and optimize the spectrum. It's just we rarely do. I guess if you want to start making reference tables or things like that, then that is something you can explore. And that's one of the strengths. The
big advantage, though, is that even though it's relative abundance, it's a very robust relative abundance. Even if you do it in different countries, it will give you the same answer. Whereas if we do it in an untargeted LCMS-based analysis, just to remind everybody, normally when we do untargeted LCMS-based analysis, we aim to finish all the analysis in one go. And then that's because there's just a lot of variability and instability in the analysis. But with NMR, you can do it in batches in different countries, of course, as long as the machinery is the same. In line with the question, there's a question: which softwares do you use? Do you use some open source platforms, or
is this for processing and informatics? So for processing, we tend to do that. It's automated in TopSpin, Brooker TopSpin. So if you're using, I assume it's probably a Brooker instrument that you're using over there, which would, I assume, come with TopSpin. I think it has to have TopSpin to run. So all the initial processing is done there. And then for the informatics, for that kind of digitization of the spectra, we tend to use in-house scripts in MATLAB, but a lot of those are now being converted to R scripts. So there's one, Torben Kimhofer has developed one called Metabolomics. I think the mate might be spelled M8.
Right. But the moment Yeah, well, the moment you have the data table as well, the table that John presented a while ago, where the columns are the bins and the rows are the observations, you can also use Metabolomics for that analysis.
Yeah, it's a bit because you have 20,000 inputs and the column names are like the spectral position, and so one peak might have sort of 20 individual dots, so it becomes a bit trickier to interpret. So yeah, so one way is to use something like Metabomate, and you can do normalization in that as well. Or you can, what some people do, which starts to move, I mean, Brian might have his own thoughts on this, it starts to move away from untargeted and more towards targeted. But what some people do is they literally go along the spectrum and they annotate everything they can. You do lose that information, but it means that you can use online tools like Metabolomics, and I see more and more people doing that. But yeah, you lose that really interesting information. Might be, you know, a really unusual metabolite that comes from the diet.
Yeah, there's a follow-up question on the lower limit of detection. I'm just curious, Maz, which institute are you affiliated with? Are you from De La Salle or?
Maybe we can allow Mas to speak. I think he's from Sydney.
As from okay, and okay, you're using NMR already in your study then. If you can give a shout out. All right, let's give it time. Okay, with regards to the biological interpretations that you have, it's very interesting because of course stunting remains a big problem in the Philippines, and there's a big interest in study, and I defer that to Jacobs to tell you more about the situation and the research interest that we have. But there's a quite interesting question here, whether it's actually a funny question to me. How does this information trickle down to communities? Do pediatricians use our results? What is your experience on this? I have my own experience, but what is your experience on it?
Yeah, yeah, those are really good questions. So the way I've experienced things is, so a lot of this work, I should have said, was primarily funded by the Gates Foundation. And so I found that they are very responsive in that when we had the nicotinamide findings, they funded that illicit trial. We also, when we initially had the tryptophan and choline findings, they put some funding behind some of those egg studies I mentioned, although they're also very quick when they didn't, I don't think they had a huge impact on growth, some of those later egg studies. And so they were stopped. Paul Kelly, actually, who this is in collaboration with, he is a gastroenterologist working in the field in Zambia. And so he does incorporate a lot of these findings. It does inform what he does, but in terms of sort of, within the communities, I think we're a step detached.
Yeah, well, you also have to understand. Yeah, John and I were not pediatricians. we are doctors, but not the right kind. I think. Yeah, but for us to do the research that we do in stunting and malnutrition, we work with a lot of pediatricians, and there are a lot of pediatricians involved. But we don't. There are people who use our results to develop new interventions and community-based, but of course we cannot speak for all pediatricians, and that's the objective. That's why we do the science that we do, to hopefully influence medicine, medical practice in a larger scale. But I'll defer to Jacobs to give us your insights into research and how can this lecture be used in our quest.
Yeah, well, perhaps I cannot. Well, the question regarding the use of these platforms in clinical practice, particularly with the pediatricians. Well, basically here in the Philippines, we are more into looking into the translational path of doing this omics research this is why my question is actually more on the translational side, because here in the Philippines, the problem on stunting is still, how do you call this, unanswered, I guess, because we continue to lag behind our targets in eliminating the problem on stunting here in the Philippines. This is why there's this big shift, I think special focus on research about stunting here in the Philippines. So maybe the question that I have now is more on the translational perspective. If well, coming from all of this research that we do in the omics platforms, building the link between the metabolomics and the metagenome, for example, at the end of this research, perhaps, can we have some, can we derive specific, like for example, metabolites or strategies wherein we can actually base our specific interventions? Like, for example, can we develop food products or formulations of products that can actually help compensate for the lacking metabolites, for example, in stunted children?
Yeah, I mean, I would hope so. Yeah, so that is definitely the goal. So I think, yeah, I think the NAD plus findings are really interesting. There's the tryptophan findings, but I think even amino acids in general. But I guess if you've got something like gut damage preventing that absorption, then does that need to be corrected first, or do there need to be alternative methods for sort of supplementing those pathways? And whether that could be the microbiome. So if you tweak the microbiome with pre or pro or symbiotics, could you improve gut health, which then allows these amino acids to get absorbed? So yeah, I would say it can inform nutritional strategies. But yeah, I think it has to be the wider picture as well. And also different times, different ages.
But if I might also add, because yeah, inspired by Natasha's work, we also tried to do we also did a longitudinal study in Gambia, and our results also pointed out the choline, in fact, as an influencer towards stunting. We're actually also happy that Science Advances also got that paper out. But what we also found was that choline, although has it could be a potential, has a potential causal link to improving growth, choline alone does not do the job. So that's why John was saying a while ago, it's very important for us to have a more holistic view, because the way we do clinical interventions or nutritional interventions right now is we're very focused on specific. We're actually looking for this magic bullet. This miracle, supplement that we can give to children, and then they will just start growing. But that's not the case, and we know that already time and time and again. And I think the use of these approaches will help us know which combinations together actually drive the growth in children. So we have, as John mentioned, nicotinamide and tryptophan. They're of course related, so it could be an entire amino acid profile that's problematic, plus choline, plus PUFA, plus all these things. So yeah, that's my long answer to the question. Gordana is here, and I see her hand is raised.
Her hand.
Yeah, so maybe you can unmute yourself.
Hi, Gordana.
Welcome back.
Welcome back.
Hi, thanks. Yeah, of course. What a great session this entire conference. Yeah, no, I just wanted to tag on to what you were saying, Brian, because I think one of the things that you see in the PAS paper is like we've plotted the individual metabolites that contribute to PAS, as John showed. But you won't see like huge differences in between the healthy and non-healthy children in those individual metabolites. But nonetheless, when you compose them into this phenome score, there are huge differences between stunted and non-stunted children. So I think this is kind of pointing towards what you're saying, that like you may not get this magic bullet one single metabolite. And this is why metabolomics is really great, because what we've identified is like, a couple of them that act in concert. that you might not otherwise be able to identify if you're just doing univariate stats or pursuing a single metabolite. That's just a plug for metabolomics right there. Go Metabolomics.
Well, amongst this crowd, I don't think there's a lot of convincing to do. I think everyone is very convinced. Do we have any questions from YouTube?
Allow me to check it.
And then we can wrap this up in the interest of time if we have there.
I think there's none.
I guess none. So with that, and yeah, in the interest of time, there's a lot more discussions that we can do, of course. And we look forward to engaging with you, John and Gordana, on our projects that we're doing in the Philippines. And yeah, I would like to of course appreciate John has been very supportive of my personal career as well. So thank you very much, John. And with that, of course, I would like to thank everyone for your attention. John, Gordana, thank you for joining. Just a reminder of what's up next. So tomorrow would be our day 8 of this webinar. We have a talk from Professor Lynn Van Hacke of Ghent University and Queen University Belfast on the gut microbiome diet axis. And then on Thursday will be our last talk, and that is on DNA adducts by Liselot Hemerick from Ghent University. And with that, I pass it back to Diane or Jacques for our wrap up. Thank you. Thank you, everyone. Maraming salamat po.
Yes, thank you. Thank you so much. Let's give John a virtual clap. Okay, thank you for that one. As a reminder, the evaluation form link is provided in the chat box. Kindly accomplish it, and all the certificate of participation will be provided starting October 21, 2020. Thank you, and see you again tomorrow for our next episodes. Bye.
Bye,
Thank you,
John.
everyone.
Thank you, Gordana.
John, Gordana, let's keep in touch. Bye.
Thanks, Brian. See you soon.